Assalamu alaikum. I'm Nida Javaid, PhD botany scholar. Today my topic of presentation is complete chloroplast genome of Farsetia hamiltonii Royle, comparative analysis within genus Farsetia, and phylogeny of Clade C of Brassicaceae. Farsetia hamiltonii is a medicinal plant commonly called freed booty or latti, belong to the Clade C of family Brassicaceae. It used to treat pain, inflammation, swelling in joints, diabetes, gastrointestinal, and infectious diseases by the common inhabitants of Cholastan Desert and other Indian regions. Reason of choosing this species due to its medicinal importance. Only one complete CP genome of Farsetia occidentalis is available on NCBI, which is insufficient to resolve taxonomic issues at species level. We reported complete CP genome sequence of Farsetia hamiltonii Royle from Cholastan Desert, Punjab, Pakistan, and performed comparative analysis with Farsetia occidentalis. Chloroplast genome, it consists of four chambers, two inverted repeat regions, IRA and IRB, one small single copy and one large single copy. It encodes for key proteins for photosynthesis and other metabolic processes. Here we go for the material and methods. We collected fresh plant leaves from the Cholastan Desert. The DNA was extracted following the method of used by Ahmed et al. It was a whole DNA extraction method. The quantity and quality of extracted DNA determined by nanodrop and 1% agarose gel electrophoresis. Alumina HiSec 2500 was used to create a complete genome shotgun by the pair and library of 150 base pair at Beijing Institute of Genomics, Beijing, China. De Novo assembly and annotations was done, quality determined by FastQC, assembled with the help of Velvet. De Novo assembled in Genus and reconfirmed in Novoplasty. Annotate using GSEC. Average sequencing coverage was determined by using BWA mapping and visualized in tablet. Circular maps was made using OGDRA. Complete CP genome submitted to the gene bank and accession number was assigned. Bioinformatics analysis, which I have done in this paper. Codon usage using Mega6, RNA editing sites prediction, PrepCP, IR contraction and expansion with IR scope, analysis of microsatellites using MESA, analysis of oligonucleotide repeats, repeater program, synonymous and non-synonymous substitutions rates. It was done with Genus and KS calculator. SNP's analysis and highly polymorphic regions was determined by making alignment in MAPT and Genus with the help of Genus. Phylogenetic analysis was done by downloading 22 species of Clade C from NCBI and aligned 23 sequences in Genus using MAPT. Phylogenetic tree was made using IQ tree and it refined using ITIL. Results and discussion. Chloroplast genome structure of Farsetia hamiltonii consists of a CP genome, complete CP genome of size 154,802 base pair. Small single copy region consists of 17,988 base pairs, large single copy 83,906 base pairs, and IRA and IRB length was 26,454 base pair in length. Total number of genes were found 132, protein coding genes 87, tRNA genes 37, rRNA genes 8, and 20 genes were repeated in IR regions. This one is the structure of Farsetia hamiltonii. It is not expanding here. I have displayed it in the other window. This one is right here. I can zoom it. These are the four regions: IRB, IRA, SSC, and LSC, and all the genes have been shown around it. Okay. Comparative analysis of Farsetia hamiltonii with and Farsetia occidentalis. Both have typical quadripartite structure comprising of one large single copy, one small single copy, and two IR regions. Both showed high resemblances in genome sizes and genome gene structure. GC content codon usage and amino acid frequencies, RNA editing sites, simple sequence repeats, and oligonucleotide repeats. Here I have showed the comparison of Farsetia hamiltonia and Farsetia occidentalis of amino acid frequencies. Highest amino acid frequencies was found of leucine. Second is RNA editing sites in Farsetia hamiltonia and Farsetia occidentalis. Highest RNA editing sites was observed in RPOC1 in both the species. Higher contraction and expansion. Here I have showed the regions of inverted repeats, inverted repeats, both the species. You may see the four junctions. Four junctions have shared the same genes in among the both species. Here is the RPS 19. You may say it. You may see the RPS 19 in the Farsetia hamiltoni like this, and DHF and DHF. All the genes which exist on the junctions are similar in both the species. Microsatellites, simple sequence repeats observed was mono in both. The location, region-wise distribution is also similar in both the species. Oligonucleotide repeats, the same sequence in both species. Transversion ratio in both species was observed, and the transversion ratio was higher than the transition substitutions. Here I have displayed the 10 highly polymorphic regions observed in Farsetia species, which can be used as markers in identifying the species. Phylogenetic tree of 23 species of Clade C, which have showed Parolinia and Mauritia as closest genus to the Farsetia. Conclusion: Farsetia, concluding remarks, Farsetia hamiltoni and Farsetia occidentalis both have similarities, which also observed similar results observed in other Clade C members of family Brassicaceae. All members of Clade C family Brassicaceae showed monophyletic origin and high resemblance with the Farsetia genus, ranging from 96 to 91%, and node of each genus was well resolved as compared to the other genera. Maximum likelihood tree toward Farsetia, a monophyletic genus closely related to the genus Parolinia with 96% similarities. The average rate of non-synonymous substitutions was recorded higher than the synonymous substitutions. We identified 10 highly polymorphic regions that could be used to develop authentic and cost-effective markers to resolve taxonomic issues and infer phylogenetic relationships among Farsetia species. Thank you so much from my side.